Good evening. Tonight we begin with something a little unseen and unceremonious. Radiation. Invisible and indifferent, it slips through air and tissue alike, and tonight we'll trace, second by second, what it actually does when it meets the human body and the fragile code inside each cell. Forget the Hollywood flashes and glowing mutations. Radiation's real work is quieter and far more precise, an arithmetic of electrons, shattered bonds, and microscopic breaks that rarely look dramatic until their consequences add up. The truth is, the moment energetic particles pass through you, the chemistry of your cells is altered. Electrons are knocked loose, water becomes chemically reactive, and DNA can be nicked or snapped. Repair systems rush in, and whether those first seconds end in recovery or lasting change is the story we're about to follow. If you're enjoying this, take a moment to like the video and subscribe, and tell me in the comments where you're tuning in from and what time it is there. I'm always fascinated by the variety of places people are watching from. Silence your notifications, take a deep breath, and let's begin. Alpha. Particles arrive like a tiny blunt battering ram. 2 protons and 2 neutrons bound together, the nucleus of a helium atom carrying a positive charge and a surprising amount of momentum. When Marie Curie and her colleagues in Paris isolated radium and polonium at the end of the 19th century, they were not simply unearthing new elements. They were exposing the world to different species of emissaries that would teach the century how matter gives up its secrets. Henri Becquerel in France saw the first shadow of that new kingdom in 1896. Wilhelm Röntgen in Würzburg had already revealed X-rays in 1895. Ernest Rutherford, first in New Zealand and then at Manchester and Cambridge, spent the 1900s and 1910s reading the behaviour of these emissaries and naming them alpha and beta, thin and thick, immediate and insistent. An alpha particle is heavy for a particle of radiation, about 4 atomic mass units, and it carries a double positive charge. Physics gives it a short fuse. A 5 me for V alpha will travel only a few tens of micrometers in tissue, losing energy densely along its short path. That dense shorthand writing of deposited energy is what physicists came to call high linear energy transfer, or LET, measured as energy loss per unit length, the keV per micrometer that tells you whether damage falls like scattered raindrops or is hammered into the wood like blacksmith blows. Beta particles are different. Lighter, fleet-footed, electrons or their antisymmetric cousins, positrons. They are the skittish couriers of radioisotopes, the beta-emitting daughters of strontium or iodine or the tritium used in laboratories once upon a time. James Chadwick would later give us the neutron in 32 in the Cavendish laboratory at Cambridge, and the world of nuclear physics would expand again, but by then people already knew that betas penetrate further than alphas. They are charged, yes, but their single elementary charge and low mass mean they scatter easily, change direction, and tend to deposit energy sparsely compared with the alpha's concentrated trail. An X-ray beam in a radiologist's room and a medical gamma source used for therapy or a diagnostic tracer both act through the electrons they set in motion. The photons themselves are uncharged and indifferent, passing through until an interaction produces a charged agent that will do the real work of ionization. Photons, the X-rays that Röntgen announced in 1895 in Germany, and the nuclear gammas made plain by Paul Villard in France in 1900, have no mass and no electric charge, and they seldom touch directly. They interact with the electrons bound in atoms by 3 principal mechanisms. In the low-energy end, the photoelectric effect plucks electrons out of shells. At intermediate energies, Compton, scattering kicks electrons and changes the photon's direction and energy. And at the highest energies, pair production briefly manufactures matter from light. The result is always the same. The photon's energy is handed off to an electron, and that electron, now energetic, becomes a small mobile source of ionization. Because those secondary electrons are produced sparsely and can travel relatively far, photons are said to have a low let in biological tissue. Their damage is widely distributed, often a single lesion scattered across large molecular fields. Neutrons play a quieter but more dangerous game. James Chadwick's discovery in 1932 made clear that neutral particles, with roughly the mass of a proton but no charge, could barrel into atomic nuclei and transfer appreciable energy through collisions. In hydrogenous tissue, human bodies are mostly water. Elastic collisions between fast neutrons and protons create recoil protons. Charged, heavy particles that look like mini alphas in their biological effect, depositing energy densely along short tracks. Neutrons also induce nuclear reactions that give off gamma rays and charged fragments, so exposure is a messy orchestra of secondaries. The physics community in the 1930s and 1940s, Enrico Fermi in Italy and later in Chicago, developing neutron sources and initiating controlled fission in 1942 at the University of Chicago's Stagg Field with CP1 on December 2nd, the Manhattan Project laboratories at Los Alamos, Oak Ridge and Hanford during World War II, transformed neutrons from theoretical curiosity into the practical protagonists of chain reactions and, unfortunately, weapons. The Trinity device over the Jornada del Muerto on July 16, 1945, and the bombs dropped on Hiroshima and Nagasaki in August of the same year were not merely historical ruptures. They were brutal proofs that different kinds of radiation, in scales and mixes, would interact with human tissue in temporally and spatially complex ways. Mass, charge and energy determine the way each kind of radiation deposits its energy. A charged particle carries a Coulomb field. As it moves, it drags that field through matter and pulls electrons out of atoms in its immediate neighbourhood. That interaction is local and frequent for heavy charged particles, many ionizations per micrometer of path length. A photon, because it carries no charge, must gamble on an interaction that first creates a charged particle. Its energy deposition is discontinuous, more sparse and can be scattered. A neutron neutral does not interact electromagnetically. Its fate is collisions with nuclei, and the energy it deposits depends on the mass ratios and cross-sections of those encounters. The cross-section for a given interaction is energy dependent, and for photons in particular, the probability of photoelectric absorption favours high Z materials at low energies, while Compton scattering dominates for soft tissue at diagnostic energies, and pair production becomes relevant at high energies. These are the rules that determine penetration depth and initial interaction. Probability in the human body. They are the logistics of the attack, and the attack begins in an eye blink that is far too fast for our biological clocks to see. The deposition of energy from an ionizing event is essentially instantaneous, from our human perspective, occurring on attosecond to femtosecond scales for the initial ionization and excitation events, and continuing into the femtosecond to picosecond range as the ejected electrons thermalize and cascades of lower energy electrons form. The physical stage of radiation action is over before a blink finishes. Those few femtoseconds write the initial ledger from which chemistry and biology must later attempt to reconcile accounts. When one shrinks the world down to a micrometer and below, the neat categories, alpha, beta, photon, neutron, blossom into patterns and architectures of ionization. If you imagine tissue as a quiet, foggy moor of water molecules studded with proteins and DNA, the passage of a charged particle carves a path. Ernest Rutherford and his colleagues in early 20th century laboratories watched cloud chamber tracks and later Wilson chambers to see these paths in a macroscopic sense. Decades later, those macroscopic shadows were resolved into microscopic tracks and their implications for biology. A heavy charged particle drives along a core, a central column of very dense ionizations where energy deposition is intense, flanked by a halo of secondary ionizations made by energetic electrons that were knocked free and sent out radially. Those energetic secondary electrons are called delta rays, little offshoots that can travel tens of nanometers to micrometers away from the main track depending on their energy, producing further ionizations along their own short journeys. The picture, at the nanometer scale, is that of a seared trail with sparks flying sideways, a primary core and a spray of delta rays that define the radiation's microscopic footprint. The Bethe formula, developed and refined in the early 20th century by physicists like Hans Bethe, gives us a quantitative sense of how charged particles slow down in matter, the stopping power dE/dx, and it ties the particle's mass, velocity and charge to how much energy is shed per unit length, William H. William. Bragg and his contemporaries had already observed the Bragg curve, the idea that charged particles deposit most of their energy near the end of their range, work that would, a century later, be harnessed deliberately in proton therapy to place dose precisely at tumour depth. The practical upshot for biology is this. A single traversing alpha particle, with its highly T, deposits a lot of energy in a micron or less. A photon, by contrast, spreads its energy more diffusely and the clusters of ionization that lead to complex lesions are less common per unit dose. Time here is intimately tied to geometry. The primary ionizations and the creation of delta rays occur within sub-nanosecond windows. The initial spatial distribution, the clustering of ionizations within tens of nanometers, sets the stage for what types of lesions will be observed seconds later. If you disrupt DNA in a way that leaves separate single-strand breaks scattered by tens of nanometers, repair enzymes can often manage the damage. Put those breaks and base lesions together in a cluster within a single helical turn or two, within a region a few nanometers across, and the result is what radiobiologists call complex damage. Multiple, closely spaced lesions that impede and confuse the cell's repair machinery. A double-strand break may arise from two closely opposed single-strand breaks on complementary strands, or it may be an immediate consequence of a single high-energy deposition that removes electrons from both strands at once. In either case, the physical geometry of the ionizations, how many, how close, how simultaneous, governs biological fate. Secondary electrons carry a statistical distribution of energies. A 100 electron-volt electron moves only a few nanometers before it is stopped. A kilo-electron-volt electron may roam tens or hundreds of nanometers. Because damage to DNA is governed by events within just a few nanometers of the molecule, the energy spectrum of secondary electrons hugely influences whether a given radiation track is likely to produce complex clustered lesions. High LET radiations, like alphas or heavy recoil protons from neutrons, produce very dense ionization cores where clustering within a few nanometers is common. Low LET radiations, like the secondary electrons from a gamma cascade, produce a sparser sprinkling of hits, many isolated single lesions, fewer clusters. The distinction matters for repair, for mutation, for cell death. All this electron drama unfolds before chemistry has had time to start complicating the stage. The most abundant molecule in tissue, water, provides the substrate for the next act. Radiation chemistry had been observed and debated since the early 20th century, but a dramatic expansion of understanding occurred alongside the wartime and post-war development of accelerators and reactors, where chemists and physicists needed to know what would happen when radiation met living or engineered materials. Water radiolysis is the shorthand for the way energy deposited into water produces a cascade of reactive species, and it is the primary route by which many forms of radiation induce biological harm. When an energetic particle or a photon transfers energy to water, it can remove an electron from a water molecule, producing H2O+ and a free electron. It can also excite a water molecule without ionizing it, producing H2O. The immediate fate of those species is rapid and predictable. The ionized water molecule is a highly reactive cation that quickly transfers its charge to nearby water molecules, generating hydronium ions and hydroxyl radicals. The excited water can split into hydrogen atoms and hydroxyl radicals. The free electrons, slowed by collisions, become thermalized and in the presence of polar solvents become solvated, captured by a cage of surrounding water molecules to form the so-called hydrated electron, EQ. All of these initial events, the creation of H2O+, H2O, EQ, OH and H, take place in the picoseconds after the primary ionization events. This is the physico-chemical stage, and it bridges the raw physics and the ensuing chemical drama. If one were to whisper the list of actors, hydroxyl radicals, hydrogen atoms, H, solvated electrons, EHQ, molecular hydrogen, H2, hydrogen peroxide, H2O2, and eventually oxygen-derived species like superoxide, O2, in an oxygenated environment, the names sound small and innocent. They are not. The hydroxyl radical is, in the chemical sense, a wild animal. Highly reactive, non-selective, and capable of abstracting hydrogen atoms from sugars and proteins, or adding to aromatic rings of bases. The solvated electron is a reductant capable of attaching to molecules and setting in motion different pathways of damage. The initial yields of these species, the so-called G values used in radiation chemistry, are modest numbers per 100 eV of energy deposited, usually on the order of a few molecules. That may sound trifling until you remember the density of deposition in a high LET track or the sheer number of events across a larger dose. Moreover, the diffusion distances of these species are short. Hydroxyl radicals, for instance, travel only a few nanometers on average before they react. They are local agents of havoc. Hydrogen peroxide, by contrast, is a stable messenger. It can diffuse far beyond the original site, survive seconds to minutes, and carry oxidative potential elsewhere. The chemistry changes quickly with time and with oxygenation. In the first picoseconds, electrons solvate, excited states decay, and the primary radicals are formed. In the tens to hundreds of picoseconds and into the nanoseconds, those radicals begin to encounter one another and the surrounding biomolecules. Recombination events reduce total radical counts, but where water breaks down near a vulnerable target like DNA, the radicals can react with the molecule's sugar-phosphate backbone or its bases. By the microsecond and millisecond scale, diffusion takes over. The more stable species like H2O2 and molecular products of radical recombination wander and can affect sites more distant from the initial track. Seconds later, chemical cascades have produced a changed redox landscape. Peroxides and oxidized thiols, altered protein conformation, and oxidized lipids that will ripple across membranes and into signaling networks. These are the seconds that turn physics into biology. If a hydroxyl radical abstracts a hydrogen from the deoxyribose sugar in DNA, the chain breaks. If it adds to a base, the chemistry of base pairing is altered and a mutation may be born. A solvated electron can reduce a base or a radical site, forming a different lesion. Oxygen acts as a treacherous ally. It reacts rapidly with DNA radicals to form peroxyl radicals, and in doing so it often fixes damage, meaning that damage which might otherwise have been chemically reversible becomes chemically permanent and thus more likely to be mutagenic when the cell tries to copy or repair. Louis Harold Gray in Britain and others in the mid-20th century formalised the oxygen enhancement effect. It is the reason why tissues with good oxygenation are more susceptible to radiation in therapeutic settings, and why hypoxic tumour regions resist treatment. The oxygen enhancement ratio for low LET radiation is typically in the region of 2 to 3, that is, oxygen can roughly double or triple the biological effectiveness of the same physical dose. High LET radiation, because it creates more direct double hits on DNA, is less subject to oxygen modulation. The spatial clustering of chemical reactions set up by the track structure milliseconds before is therefore a determinative feature. Consider two scenarios. 1. A gamma photon produces a secondary electron that wanders and produces a handful of widely separated single-strand breaks and base modifications. Cellular enzymes, nucleases, glycosylases, polymerases come to work and over minutes to hours patch and repair many of these lesions. 2. An alpha particle rips straight through the nucleus and deposits a dense cluster of ionizations within a region the size of a few turns of the DNA helix. Hydroxyl radicals and direct ionizations create multiple strand breaks on both strands, crosslinks, and oxidized bases, all within a single, locally crowded molecular neighborhood. Repair in that case is fraught. The typical efficient pathways, base excision repair for singular base lesions and single-strand breaks, non-homologous end-joining or homologous recombination for double-strand breaks are challenged by the proximity and diversity of lesions. Misrepair can lead to translocations, failed repair leads to cell death. The cellular fates that follow, apoptosis, senescence, mutation, or survival, are the downstream narratives of that first and furious second. There is historical poetry to this chain of understanding. Hermann Joseph Muller's experiments with X-rays on Drosophila in 1927 established that radiation could elevate mutation rates, tying what we now think of as microscopic chemical lesions to the macroscopic changes in heredity. In the mid-20th century, the growing discipline of radiation biology, with figures like Louis Harold Gray in Britain and the extensive wartime research in the United States and Europe, built the conceptual bridge between particle physics and cellular response. The marshalling of large laboratories, University of Chicago under Fermi's Committee for the Chain Reaction, Los Alamos under Robert Oppenheimer during the Manhattan Project, the Atomic Bomb Casualty Commission, established in 1947 to study survivors in Hiroshima and Nagasaki, and later succeeded by the Radiation Effects Research Foundation, meant that these were not just lab curiosities. They became matters of public policy, of medicine, of ethics, and of national logistics. It is important to picture the seconds after exposure not as a single simple event, but as a layered progression. The physical interactions write down a pattern of ionizations in femtoseconds. The physico-chemical stage in picoseconds reveals a small army of radicals. The chemical stage, picoseconds to seconds, watches radicals attack, recombine, and convert into longer-lived molecular products, and only then, after seconds and minutes, do cellular signalling pathways enact responses that take minutes, hours and days to complete. In practical terms, what matters at the second scale is not just how many electrons were liberated, but where they were liberated, and what the solvent environment and local chemical milieu do with those electrons, and the radicals that form around them. Diffusion distances and reaction rates rule that second. A hydroxyl radical may act almost in situ, altering a nearby base or fragmenting a sugar ring. A solvated electron may be scavenged by oxygen if present, creating superoxide and propelling the chemistry into a different oxidative direction. A glutathione molecule or a thiol-containing protein may quench radicals, limiting early damage. But antioxidants come at a cost. They are consumed. The balance of production and scavenging in the seconds after irradiation shapes whether the cell will deal with a transient insult or be launched into a chronic oxidative state. Hydrogen peroxide, born in the aftermath, is a slow messenger. It diffuses and can permeate membranes, turning a localized radiation event into a broader redox signal that may activate kinases, transcription factors and stress responses. Different kinds of radiation therefore not only set different initial conditions but skew the seconds-by-second chemistry that follows. In tissue where water dominates, the indirect mechanism, radical attack, can account for upwards of 2/3 of DNA damage from low LE radiation, a reality that early radiation chemists and biologists discovered with painstaking experiments in the middle of the 20th century. High LET radiation tilts the balance back toward direct ionization of DNA itself, making geometry and clustering more decisive. Neutrons in particular have complicated reputations because their interactions produce a melange of secondaries: recoil protons that act like charged heavy particles, gamma rays and residual nuclear fragments, so their biological effectiveness tends to be higher than an equal dose of photon radiation. This had practical and political significance in the Cold War. Dosimetry for neutrons was harder, and particle mixes in fallout complicated epidemiology. You can imagine, if you wish, a single ionizing event as the opening line of a story whose chapters are written in different languages. Physics first, then chemistry, then biology. The initial sentence is terse and fast. The middle chapters are messy and full of argument. And history has been the slow and persistent librarian of this story. Early radiologists in the United States and Britain learned the need for shielding the hard way in the first decades of the 20th century. Marie Curie's own work, her founding of the Radium Institute in Paris and her tireless wartime service bringing mobile X-ray units to field hospitals, created both therapeutic opportunity and exposure tragedy that taught caution. Later structured studies, Hermann Muller's genetics work in 1927, the wartime research into radiation sickness, the post-war epidemiological efforts on survivors in Japan, connected the dots between microscopic lesions and population outcomes. Technological advances, accelerators, reactors, and later Monte Carlo computational methods developed at national laboratories gave scientists the ability to simulate track structures and test hypotheses about microscale clustering and its consequences. In the hush of a laboratory late at night, a physicist can run a Monte Carlo track simulation and watch, in plotted numbers, the same pattern a cloud chamber once hinted at: a primary ionization, a delta ray branching, a small cluster of energy dumped into a region the size of a few DNA turns. A radiochemist can measure transient absorption spectra and see the solvated electron appear and fade in picoseconds. A biologist can stain for gamma H2AX foci and count the double-strand breaks that seconds to minutes later mark places where the cell has recognized severe damage. The arc from the initial deposition of energy to biological consequence is long and interdisciplinary, and it has been constructed by laboratories in Cambridge, Paris, Chicago, Los Alamos and Tokyo, among many others, places where careful people kept records, refined measurements and argued across disciplines about what really mattered. In those first seconds, the human body is both inert and hyperactive. Inert because the fundamental physics is done, hyperactive because chemistry rushes in to fill the void. The molecules are invisible, but their reactions are as certain as a ledger entry. Electrons captured, radicals formed, peroxides accumulated. Some molecules are destroyed outright. Others are modified in ways that will be recognized and mended or misrecognized and misrepaired. If one were to give this scene a taste, The initial moments are metallic and bright, sharp with the smell of ozone in ambient air and with the knowledge that at the molecular level, bonds are being bent and broken in ways no biological evolution ever intended them to be broken. The smell is not literal inside cells, but the world of medicine has learned that radiation leaves signatures, chemical fingerprints that doctors and biologists can detect later, in the form of lesions, mutations, and in the long view, cancer risk or tissue failure. A modest dry thought to close this segment. For a century we have been learning not only that radiation is powerful, but precisely how it is powerful. The pioneers, Röntgen in Würzburg in 1895, Becquerel and the Curies in Paris in the 1890s, Rutherford in Manchester and Cambridge in the 1900s, Chadwick at Cavendish in 1932, Fermi in Chicago in 1942, Oppenheimer and the wartime teams at Los Alamos in 1943-45 provided the tools. Later generations, radiochemists and radiobiologists in the decades after World War II, in labs from London to Tokyo, filled in the fine print, the picoseconds and nanometers that determine whether a cell will live, die, or change. The human body reacts to ionizing radiation on timescales that span femtoseconds to seconds and beyond. And the story of those first seconds is a layered tale of particles and tracks, of fast electrons and delta rays, and of water, humble, everywhere, splitting into a storm of radicals that will decide, in tiny chemical votes, the fates of molecules and, by extension, of cells and organisms. It is a quiet, relentless calculus, the physics of the very small making moral demands on the very large, and it is a tale whose details we have learned patiently through the laboratories and hospitals and, yes, the unfortunate experiments of history, one small measured observation at a time. The first seconds after an ionizing particle slices through a cell are a small savage theatre of physics and chemistry, a place where one measured beam or stray photon can turn the quiet grammar of a genome into a scattering of broken letters. Imagine, if you will, an invisible bolt, an X-ray, a gamma, an alpha particle, passing through the nucleus like a messenger with a temper. It does not knock politely, it robs electrons, it excites bonds, it splinters sugar phosphate backbones. When the particle hits DNA directly, it can pluck an electron from a base, or cleave the backbone in a single high-energy strike. That is direct ionization. The molecule itself bears the blow, atoms suddenly lacking the electrons that once made their chemistry sensible. These are events of incomprehensible speed, femtoseconds to picoseconds, but their consequences arrive in the human scale of seconds and minutes as lesions that repair enzymes must reckon with. If the particle does not strike DNA head-on, it can be no less lethal. The nucleus is mostly water, and water is an obedient, if untrustworthy, ally. Ionizing radiation splits water molecules into reactive fragments in a process called radiolysis. In the first trillionths of a second, neutral water gives rise to hydroxyl radicals, hydrated electrons and molecular hydrogen. Within microseconds, those radicals roam, colliding with bases and backbones, oxidizing guanines, cleaving sugars, gouging out bases. This indirect attack, chemistry initiated by water, accounts for a large fraction of biological damage from low linear energy transfer radiation such as X-rays and gamma rays. High linear energy transfer radiation, like the alpha particles released by some radionuclides or the heavy ions used in particle therapy, lays down energy in dense tracks and causes clusters of ionizations that mix direct hits and congregations of radicals in the same tiny patch of DNA. The difference matters. How energy is delivered, sparse and random or concentrated and clustered, shapes the topology of damage, and that topology shapes the cell's fate. Within those first seconds, the assault creates a catalogue of molecular injuries that a cell's repair crews will spend minutes, hours, and sometimes its entire lifespan trying to correct. Oxidized bases are common early signatures. Guanine becomes 8-oxoguanine, a lesion that likes to mispair and seed mutations. Thymine can become thymine glycol, or other bases can be chemically altered so that the base no longer fits comfortably into the double helix's code. The sugar phosphate backbone can be nicked. Single-strand breaks, the kind of scissoring that, though often repairable, becomes treacherous if opposing nicks are close by. The glycosidic bond between base and sugar can be ruptured, leaving an apurinic or a pyrimidinic site, an abasic place where the base is gone and the code shows a hole rather than a letter. Enzymes that later try to read or copy that region can stumble. inserting wrong letters, skipping or stalling. Within seconds, and sometimes by direct collision, double-strand breaks appear. Both strands severed, a cliff of missing continuity across the genetic script. Less frequent but harder to undo are DNA-protein crosslinks, when radicals glue proteins to DNA. When those proteins are historic scaffolds, histones, polymerases, transcription factors, the structure becomes fettered. The cell cannot simply slide a repair patch into place. Then there are clustered lesions, the molecular equivalent of concentrated artillery fire. Radiation does not always make one isolated wound. Often it inundates a few dozen bases with multiple lesions, oxidized bases, a basic site's nicks, and sometimes a direct double-strand break, occurring within a few dozen base pairs. Think of a short street in a village riddled with broken windows, uprooted stones and burned door frames. Repair crews cannot simply cordon off a single house and begin. They must wrestle with the debris interacting in unpredictable ways. Nanoscale clustering is the key technical problem. If two single-strand breaks occur on opposite strands a few bases apart, the very act of repairing one can convert the other into a double-strand break. If a base excision near an abasic site is processed while an adjacent strand bears a nick, the result is again a double-strand break. Enzymes working in cramped quarters can collide, trim back more DNA than intended, or leave gaps that later give rise to deletions, insertions, or chromosomal translocations. Chromatin, the spool-like arrangement of DNA around histone proteins, adds another layer of difficulty. A lesion buried inside tightly packed heterochromatin is less accessible. Enzymes stumble on nucleosomes. The same physical proximity that creates clustered chemistry also denies access to the architects who could fix it cleanly. History, as historians like to say, is written in consequences, and the story of how radiation tears and distorts genomes is stitched to key figures and laboratories across the 20th century. In 1927, Hermann J. Muller, in the United States, showed that X-rays increase mutation rates in fruit flies. That was the first clear line between radiation and heritable change, and it earned him the Nobel Prize in 1946. In the decades that followed, the Manhattan Project, although chiefly concerned with building weapons, also seeded a science of radiobiology. Labs in the United States and Britain set up to understand exposure risks and the logic of damage and repair. The discovery of DNA's double helix by Watson and Crick in Cambridge in 1953 gave these observations a molecular scaffold. Suddenly there was a clear substrate, the double-stranded helix, whose integrity could be measured, assaulted, and understood. Later studies of survivors in Hiroshima and Nagasaki, conducted under the auspices of the Atomic Bomb Casualty Commission, and later the Radiation Effects Research Foundation in Sen, onward tied cellular events to epidemiology. The microscopic ruptures that happen in the first seconds cascade into demographic patterns decades later. Scientists in the 90s and early 2000s dissected the physics and chemistry in exquisite detail. They mapped ionization tracks, measured radical lifetimes, and defined clustered damage as the hallmark of high-LET exposures. Clinicians and physicists in Japan and Germany built heavy ion therapy centres, Chiba and Heidelberg among them, intent on delivering dense, clustered lesions to stubborn tumours. The therapeutic aim was to exploit what makes some damage hard to repair. If the first seconds are the realm of physics and chemistry, the next interval, the seconds to minutes after insult, is the theatre of alarms and messengers. DNA damage sensing and signalling begin almost immediately because a living cell cannot afford dithering. The first responders are molecular sensors, enzymes and complexes that recognize breaks, nicks, stretches of single-stranded DNA, and perturbed chromatin. Poly-ATP ribose polymerase 1, PARP1, has a front-row seat for single-strand breaks. It binds within seconds, wraps the region in branched PR chains, and recruits scaffold proteins like XRCC1 and DNA ligase 3. PRP1's activity is rapid, and in its own strained metaphors, noisy. It writes a sticky sugar code that helps magnetize repair proteins to the scene. Double-strand breaks call out a different set of sentries. The mRN complex, MR11, RAD50, and NBS1 acts like a three-man reconnaissance unit sensing blunt-ended double-strand breaks and tethering ends. Q70, Q80 binds the open ends of DSBs with a mechanical intimacy, like gloved hands cupping a gap, and in so doing it recruits DNA-dependent protein kinase catalytic subunit, DNA PK-CES. which activates local processing enzymes. Above these local sensors stand the master kinases, ATM and ATR, members of the phosphatidylinositol 3 kinase-related kinase family. ATM, named for the genetic disease ataxia telangiectasia, where patients bear mutations and an exaggerated radiosensitivity, is recruited to DSBs in part by mRN. It autophosphorylates and launches a phosphorylation cascade that modifies dozens of substrates across chromatin. ATR senses stretches of single-stranded day coated with replication protein A, RPA, a signature left by resection at breaks or stalled replication forks, and it partners with ATRIP to phosphorylate its own repertoire of targets. Within seconds to a few minutes, the nucleus is awakened. Histone H2X, one of the core histone variants, becomes phosphorylated on serine 139 to form GH2X. GH2X is effectively a chemical flag planted around a break. In mammalian cells, the flag can extend across megabases, a broad siren visible by microscopy as phoci. That spreading is not mere decoration. It amplifies the alarm, recruits mediator proteins such as MDC1 and 53BP1, and organizes a repair compartment. The dynamics of signaling matter as much as the identity of the signals themselves. Parylation by PRP1 is immediate, short-lived and reversible. Phosphorylation by ATM and ATR can set up a more extended domain of altered chromatin and long-range recruitment. Checkpoint kinases, CHK1 and CHK2, become phosphorylated in turn and enforce temporary halts to the cell cycle, giving the repair machinery precious time. P53, the guardian of the genome, receives modifications that stabilize it and enable it to transactivate genes like P21, which arrest the cell cycle, and PUMA and BAX, which can steer the cell toward programmed death if repair is impossible. The amplitude of the signal, the number of phosphorylated substrates, the breadth of GEH2AX domains, the extent of parylation, affects whether a cell pauses, repairs, or self-destructs. A small local lesion produces a whisper. A forest of clustered lesions produces a wail. And timing is crucial. Delayed or blunted signaling can permit replication to proceed across damaged templates, fixing errors into mutations. Overblown signaling can precipitate apoptosis, a tidy but irrevocable choice. For a historian of the cell, the mid-20th century to the present is a catalogue of how scientists illuminated these alarms. In the late 90s, researchers identified GABU-H2X as a rapid and robust marker of double-strand breaks. The subsequent decades mapped out the choreography of mRN, ATM, ATR, and the mediator proteins that gather at foci. The isolation of ATM in the mid-1990s, as the gene mutated in ataxia telangiectasia, revealed why those patients are radiosensitive and cancer-prone. It also pointed to the kinase's centrality in orchestrating responses and imposing checkpoints. The implications were not merely academic. The Radiation Effects Research Foundation's long-term studies of Hiroshima and Nagasaki survivors linked early molecular events to later cancer risks, showing that the small lesions and signaling failures we discuss are not quibbles of molecular detail, but determinants of population health. If the first minute is alarm, the minutes to hours after exposure are the hard work of repair. Here the cell calls specialized crews, pathways that recognize classes of lesion and choose surgical or expedient fixes. Base excision repair, BER, is the workhorse for small non-helix distorting lesions like oxidized bases and abasic sites. DNA glycosylases, each evolved to recognize particular bad neighbors, flip damaged bases out of the helix and cleave the bond between base and sugar. AP endonuclease then cuts the backbone at the abasic site and polymerase V fills the gap, followed by ligation by ligase 3 in complex with XRCC1. BER can be fast and accurate. It is the cell's routine maintenance crew, cleaning up oxidative detritus that accrues under ordinary metabolism. Thomas Lindahl, whose work in the 70s and beyond illuminated the intrinsic instability of DNA and the need for basal repair, would later be honoured among others for elucidating these processes. Nucleotide excision repair, NER, handles bulky, helix-bending lesions, those created by ultraviolet light or large chemical adducts. In global genome NER, a surveillance complex including XPC recognizes helical distortion. In transcription-coupled NER, Stalled RNA polymerase attracts the machinery. TFIH's helicase subunits open the helix, XPA verifies, and a dual incision removes a 2430 nucleotide oligonucleotide containing the lesion. Polymerase then fills the gap. NER is more elaborate and generally slower than BER, taking tens of minutes to hours depending on chromatin context, but it is robust and capable of restoring original sequence when the template is intact. Single-strand breaks, though superficially simple, are triaged by PAR-P1 and XRCC1, which coordinate short-patch repair or long-patch alternatives depending on the context and the size of the gap. PRP1's rapid binding not only recruits repair proteins, but also signals to replication machinery that a fork might need to wait. That swift local response can be a lifesaver. It is also the therapeutic target behind PARP inhibitors. Decades into the molecular era, clinicians learned to weaponize this vulnerability. Tumours with defective homologous recombination, BRCA1 or BRCA2 mutants, are exquisitely sensitive to PARP inhibition because they cannot repair the additional single-strand lesions that accumulate, converting them into lethal double-strand breaks. That strategy, synthetic lethality, was not obvious in 1927, but it sits squarely on the understanding of seconds to hours dynamics. Double-strand breaks demand a choice, and that choice is the fulcrum on which mutation and survival teeter. Non-homologous end-joining, NHEJ, is the rapid responder. Q binds virtually within seconds and protects and aligns ends. DNA PKCs is recruited, and end-processing enzymes like Artemis trim ragged. Ends, before a ligase complex composed of XRCC4, ligase 4, and XLF seals the nick. NHEJ is active throughout the cell cycle and is the predominant DSB repair pathway in G1. It is fast. It is also blunt and sometimes imprecise. When ends are processed, nucleotides may be lost. Small deletions are common. NHEJ is also co-opted physiologically for immune diversification, VDJ recombination in lymphocytes, so its existence is both creative and dangerous. Homologous recombination, HR, is the high-fidelity alternative, a kind of surgical reconstruction that uses an intact sister chromatid as template. HR requires end resection, generating single-stranded DNA that becomes coated with RPA. Rad51 replaces RPA to form a nucleoprotein filament that invades the sister chromatid and copies the correct sequence. HR is mostly available in S and G2 phases when a sister chromatid exists, and it takes longer, hours rather than minutes. BRCA1 and BRCA2 are central to this process. Mary Claire King and others mapped BRCA1 to chromosome 17 in 1990, and subsequent work revealed how these proteins facilitate Rad51 loading and preserve genomic fidelity. When HR is crippled, cells rely more on NHEJ and error-prone alternative end-joining pathways, increasing genomic instability and cancer risk, an insight that transformed both genetics and oncology. Between these poles lie alternative end-joining pathways, microhomology-mediated end-joining, polymerase theta-mediated end-joining, and other opportunistic processes that use tiny stretches of homology to stitch ends together. These are more error-prone and can produce deletions, tandem duplications, and the chromosomal rearrangements that genomes remember in the form of oncogenic fusions. The competition among pathways is not egalitarian. It is mediated by regulatory proteins. 53BP1 promotes NHEJ by protecting ends from resection, while BRCA1 promotes resection and channels breaks toward HR. Chromatin state plays its part. Compacted heterochromatin slows repair and may require ATM-dependent KAP1 phosphorylation to relax the region. Breaks in repetitive DNA pose particular challenges because mispairing can lead to deletions and transpositions. The interplay of kinetics and choice becomes clearer when one thinks of minutes to hours as a logistics problem. A cell struck in early S phase faces a different calculus than one in G1. A stalled replication fork confronted with a single-strand break risks converting that lesion into a double-strand break. The repair options narrow and the urgency intensifies. In these hours, the cell evaluates supply, availability of sister chromatids, presence of repair factors, chromatin accessibility, and demand, number, and complexity of lesions. If signal amplitude is high, checkpoints arrest the cycle and bring in heavy machinery, If the damage is overwhelming or the signalling machinery is blunted, the cell may enter senescence or undergo apoptosis. Mutation arises when repair is imperfect or bypassed. A base mispaired, an insertion fixed where a deletion should be, a translocation joining non-homologous chromosomes. These molecular errors accumulate and are the raw material of evolution and of cancer. History supplies stern reminders. The long-term follow-up of atomic bomb survivors showed increased leukemias and solid cancers in the decades after exposure, reinforcing that minutes and hours of molecular failure translate into lifetimes of increased risk. Cytogeneticists in the mid-20th century observed chromosomal aberrations, broken fragments, dicentrics, translocations in blood lymphocytes after radiation. These observations were the bridge between molecular lesion and epidemiology. Later observations of therapy-related leukemias in patients treated with radiation or certain chemotherapies exemplified the price of imperfect repair. The very interventions designed to kill tumours can perform off-target edits in genomes that later manifest as secondary malignancies. The modern oncologist reads all of this as trade-offs between dose, fractionation, and the type of radiation. Photons, protons, carbon ions. Their differing LETs and resultant lesion complexity influence tumour control and normal tissue toxicity. Even as minutes turn to hours and repair pathways complete their work or fail, cells may adopt permanent strategies. Senescence, a stable arrest accompanied by inflammatory signalling, is one way the tissue isolates risk. Apoptosis removes irreparably damaged cells. Unchecked survival of misrepaired cells seeds mutation and clonal selection. Therapeutic strategies exploit these outcomes. PARP inhibitors are lethal to HR-deficient tumours. Inhibitors of ATM or DNA PKCs can sensitize tumours to radiation. Heavy ion therapy aims to produce clustered lesions that overwhelm repair capacity in cancer cells. Each strategy is a tactical decision on a battlefield defined by nanometers and seconds, but with casualties counted in years. The first seconds then matter because they set the geometry of damage. Direct ionizations gouge and strip, indirect radicals oxidize and nick, clustered lesions confound cleanup crews. The next minutes are the heartbeats of the alarm system where PR, P1, mRN, Q, ATM and ATR send phosphorylation and parylation dispatches across chromatin, calling in mediators and enforcing cell cycle halts. In the hours that follow, the surgical choices of BER, NER, NHEJ and HR, modulated by cell cycle position and chromatin structure, determine fidelity and survival. Sometimes the cell succeeds beautifully, erasing the incident as if nothing happened. Sometimes the system repairs but alters, leaving a mutation that will be read by future polymerases. Sometimes the damage is too complex, and the cell accepts its fate, joining the natural tally of death. And sometimes, as history has shown, from Muller's fruit flies to the physicians following survivors in Hiroshima and Nagasaki, those molecular choices ripple outward to affect generations. There is a peculiar kind of irony in this microscopic drama. The mechanisms that evolved to protect us, robust, vast, and often ruthless, are the same mechanisms that, when imperfect, generate the very changes that can give rise to malignancy. Cells are frugal and clever. They patch, trim, copy, and sometimes cut corners to survive. The historian in me notes that the laboratories and clinics that have documented these processes, from Curie's Radium Institute in Paris at the turn of the 20th century through mid-century radiobiology projects in the United States and Britain, to late 20th century molecular genetics in Cambridge and the specialized ion beam centers in Chiba and Heidelberg, have observed the same fundamental truth at different scales. In the hush after the storm of the first seconds, the cell's alarm system rings, crews assemble, and choices are made. Those choices, like the dispatches across wartime communications rooms or the strategic deployments of supply lines, are exercises in triage and priority. They are exercises in history written in nucleotides. And so, as you imagine the tiny invisible skirmishes that follow a radiation track, remember that the immediate damage is both chemistry and history, physics and policy. The first seconds tell us what has been broken. The next minutes tell us who heard the alarm. The hours tell us who fixed the breach and who was consigned to memory. Science, like a careful chronicler, has mapped this timeline with growing clarity. Direct and indirect attacks in the first seconds, molecular sensors and phosphorylation waves in the next moments, and a disciplined competitive recruitment of repair pathways in the following minutes to hours. The story is neither wholly triumphant nor wholly tragic. It is instead a landscape of decisions at scales we can barely see, decisions that in aggregate have reshaped lives and guided how societies use and regulate radiation in medicine, industry and warfare. History took note when those decisions, made in molecules and in ministries, produced consequences. The sleepless historian watches calmly as cells choose, day after day, to mend or to surrender. When repair fails, the quiet choreography of enzymes and templates collapses into improvisation, and the first small betrayals can become a long, patient conspiracy. Picture a double-strand break as if someone had cut a rope ladder in the middle of a ship's hold. The two sides flap and several crews rush in. Some will follow a blueprint, homologous recombination, the careful work done only should when necessity is present to guide the seam. Others will act with blunt instruments, non-homologous end-joining, the fast, pragmatic fix that knots loose ends together and sometimes trims the frayed rope. Both are necessary. Both can fail. The history of our understanding begins with people like Hermann J. Muller, working with Drosophila in the 1920s in the United States, who showed that X-rays increased mutation rates, an early, uncomfortable proof that radiation left a literal record in heredity. He won the Nobel Prize in 1946 for that work, and his cautionary voice about genetic damage echoed into the public health responses that followed Hiroshima and Nagasaki in August 1945, and later into the painstaking epidemiology of the Atomic Bomb Casualty Commission in 1947, the body that would evolve into the Radiation Effects Research Foundation in 1975. Those were not abstract risks on a blackboard. They were numbers registered on the bodies of many, and the data would painfully trace the slow arc from molecular scars to disease decades later. Start with the small things. A single ionizing event, a high-energy photon or a tiny charged particle, can strip an electron, nick a base, create an oxidized site. Most of those lesions are the province of base excision repair or nucleotide excision repair. Quiet, efficient tradespeople that clip, replace, ligate. But even the small fixes are opportunistic. When a polymerase meets an altered base, it may stall. There are specialized polymerases, translesion synthesis enzymes, that can step in and lay down a nucleotide opposite a chemically altered base. They do their best. They do not consult the original manuscript. The result can be a single incorrect letter in the genetic text, a point mutation. That subtle alteration is easy to dismiss. It is a typo. But in 1927, Muller showed that such typos multiply under radiation, and in the decades since, every generation of geneticists, molecular biologists, and epidemiologists has seen that a handful of such typos, if they land in the wrong place, are enough to begin a malign conversation within tissue. Then there are deletions, the pages torn from the book, when the cell cannot precisely align ends, or when resection chews back DNA to reveal microhomologies, the rejoin will sometimes leave a gap. Microhomology-mediated end-joining, an alternative pathway, uses small patches of matching sequence to guide reconnection, but at the cost of losing the intervening paragraphs of code. The result is a shorter sentence, a gene missing an exon, a regulatory element gone. The machinery that normally reads those genes stumbles. Protein products may be truncated, mislocalized, unstable or gone entirely. A tumour suppressor knocked down by a deletion no longer says stop. The pace of cell division can quicken in a tissue that was once orderly. Much more dramatic are translocations, whole phrases lifted and recombined. Imagine two broken chromosomes mispaired in the frantic repair. The end of chromosome 9 stuck to the end of 22, the consequences invisible at the time of stitching. Cytogenetics made this visible. In 1960, Peter Nowell and David Hungerford in Philadelphia found a consistent abnormality in chronic myelogenous leukemia, a tiny chromosome that was later called the Philadelphia chromosome. In 1973, Janet Rowley showed that it was not a mysterious small extra piece, but a translocation, an exchange between chromosomes. The fusion gene that followed, BCR-ABL, creates a protein with new, pathologic activity, a molecular engine that drives uncontrolled growth. That translocation is a concrete example of how two misrepaired breaks, each perhaps innocuous if resolved with their proper mates, can together set a cell on a trajectory toward malignancy. Radiation, by scattering breaks across the genome, increases the probability that two breaks will be out of place at the same moment. It is worth remembering that our chromosomes are not neat, isolated islands, but a crowded archipelago folded into the nucleus, regions carrying different propensities to touch one another. In the crowded chaos of repair, distant ends can be accidentally fused. After Chernobyl in April 1986, investigators in Belarus and Ukraine noted a striking increase in papillary thyroid carcinoma in exposed children. Many of these tumours bore RET-PTC rearrangements, fusions of the RET proto-oncogene, with partner sequences, a molecular echo of breakage and misjoining. Yuri Nikiforov and colleagues, among others, documented these associations in the 90s, tying a historical environmental catastrophe to a specific pattern of genomic rearrangements in victims who were children at the time of exposure. A single misrepaired double-strand break can do more than cause an isolated error. Barbara McClintock, working quietly with Mays in the 1940s, described breakage-fusion bridge cycles. If a chromosome loses its telomere, the natural cap at the end, the exposed end can fuse with another broken end during mitosis. When the fused chromosome tries to segregate, it can break again, and the cycle repeats. The result is a chaotic amplification and loss, a cascade of rearrangements that reshapes large swathes of the genome. In human cells, radiation-induced telomere shortening or blunt breaks near telomeres can seed such cycles. Years later, researchers would find some tumours with evidence that massive rearrangement occurred not gradually, but in a single catastrophic episode, a phenomenon described in 2011 as chromothripsis. That term, chromothripsis, literally chromosome shattering, speaks to the speed and violence with which genomic architectures can be remade when repair goes wrong. Micronuclei, small satellite nuclei containing fragments or whole chromosomes excluded from the main daughter nucleus after mitosis, became suspects. Inside a micronucleus, DNA replication is sloppy, and exposed DNA can be pulverized and stitched back together haphazardly, producing tens to hundreds of rearrangements in a single cellular event. Micronuclei also serve as a visible flag for genomic instability. Cytogeneticists have long used them as biomarkers. A fragment that didn't make it into the daughter nucleus spends an anxious cell cycle in the cold. When it replicates misaligned, when repair tries aggressively and wrongly to glue it back, the future cell may have an entirely new karyotype. Karyotype changes are not merely chaotic, they are selectable. In the decades since Theodore Boveri first suggested in the early 20th century that chromosomal abnormalities might lie at the heart of cancer, we have watched selection act on the variants that misrepair produces. Some rearrangements are lethal to a cell. Others remove breaks, activate accelerators, change how a cell senses its neighbours. The body is a Darwinian landscape at cellular scale. In tissues with ongoing cell turnover, the misrepaired cell that survives, divides, and finds itself slightly better at proliferating or avoiding death will leave descendants. Those descendants accumulate further missteps, and a clonal population can emerge. The Radiation Effects Research Foundation would, in the post-war decades, map increased leukemias and other cancers in Japanese survivors. The temporal pattern, the lag of years between exposure and disease, is exactly the interval needed for a misrepaired lesion to be selected for and expanded into a clinically manifest malignancy. There is irony in the cell's coping strategies. Some are noble, some are tragically naive. In the hours and days after a genotoxic insult, the cell convenes a council. Should we stop, repair, and live humbly, or should we die with ceremony? The choices are classical. Programmed cell death, or apoptosis, was named in modern terms by Andrew Willey, John Kerr, and Alistair Curry in 1972. Morphologically, it is neat, a tidy folding of chromatin. The packaging of cellular contents into vesicles for cleanup spares the tissue the inflammatory frenzy of necrosis. Apoptosis is common in radiosensitive populations. Lymphocytes and the rapidly dividing progenitors of the hematopoietic system tend toward this fate. It is a swift, sometimes elegant response to severe DNA damage. The conductor of that decision is often the p53 protein, the guardian of the genome, from whose discovery in 1979 by Lane and Crawford, and whose later positioning as a tumour suppressor injected the literature with a moral clarity. Intact p53 pushes injured cells to either repair and arrest or to die. When p53 is mutated, a cell can tip towards survival despite harbouring dangerous errors. That survival is, from the body's perspective, dangerously promiscuous. Not every cell chooses apoptosis. Some go into senescence, described by Leonard Hayflick in 1961 as a limit to divisional potential. Senescent cells remain alive, metabolically active, but permanently withdrawn from the cell cycle. Radiation can accelerate senescence. These cells exist like shut shops on a busy street, windows dark, but their windows leak. Senescent cells secrete an array of signals, cytokines, growth factors, proteases, a behaviour we call the senescence-associated secretory phenotype, or SASP. Judith Campisi and colleagues over the past few decades have shown how ASP turns senescent cells into chronic fire starters, sustaining local inflammation, attracting immune cells, and remodelling tissue architecture. It is a compromise. The organism prevents a potentially dangerous cell from proliferating, but it pays with persistent inflammation that can impede regeneration and promote malignancy in neighbours. Autophagy, the cell's self-eating, occupies a liminal role. Christian de Duve observed lysosomes and the concept of intracellular digestion in the mid-20th century. Later, Yoshinori Osumi would be recognised in 2016 for clarifying the genetic control of autophagy. After irradiation, cells may initiate autophagy to recycle damaged organelles and survive metabolic stress. Sometimes autophagy is a survival mechanism, buying time for repair. Sometimes it is a route to cell death, a slow starvation-like demise when the damage is too great and the cell's metabolism collapses. Then there is necrosis and mitotic catastrophe, both blunt instruments. Necrosis happens when energy collapses and membranes rupture, spilling contents and provoking a howl of inflammation. Mitotic catastrophe follows a different folly. The damaged cell insists on dividing, and in mitosis the unprepared genome missegregates. The result is a giant cell with multiple micronuclei, a polyploid circus tent that cannot sustain life. It often ends in delayed death, through mechanisms that blur into apoptosis or necrosis. Importantly, the balance among these fates is not random. It is determined by dose, cell type, and genetic context. A lymphocyte exposed to a few greys will undergo apoptotic death within hours. A neuron, rarely dividing, can endure more immediate DNA damage without a fatal mitosis. Its clinical fate may be slow dysfunction rather than sudden loss. Tumour cells, notorious for p53 mutations and failing checkpoints, are prone to mitotic catastrophe or to surviving with rearranged genomes if their damage response is compromised, an uneasy fact therapeutically useful in radiotherapy, where the goal is to push malignant cells into catastrophic division, but also a peril in which surviving tumour clones emerge even more unstable and aggressive. Dose is the blunt scalpel. A whole-body dose of a few greys is typically the realm where hematopoietic failure becomes clinically relevant. The marrow, with its rapidly dividing progenitors, is exquisitely vulnerable. In Hiroshima and Nagasaki in 1945, among the many tragedies was the collapse of the bone marrow compartment in those closest to the blasts, leading within weeks to infections, bleeding, and the inexorable mortality that the world had not yet learned to treat. Above a certain higher threshold, the gastrointestinal tract succumbs. Loss of crypt stem cells in the intestinal lining leads to denuded mucosa, a broken barrier, fluid loss, and systemic infection, the classical radiation-induced GI syndrome. These are not mere textbook categories. They are the categories that guided triage plans developed in the years following nuclear detonations and accidents. The triage plans crafted by physicians and logisticians who knew that at certain doses supportive care could coax recovery, while at others the organ systems would fail no matter how many hands were turned to help. A cell's genetic context is decisive. Cells with intact cell cycle checkpoints, functional p53, and competent homologous recombination will often favour repair and arrest, taking a sober day or two to fix the genome. Cells missing these options, BRCA1 or BRCA2 mutant cells for example, will find homologous recombination is impaired and rely more on error-prone pathways. That predisposition to misrepair explains why certain genetic syndromes, like ataxia-telangiectasia, defects in ATM discovered across the late 1980s and 90s, are radiosensitive. The patients have impaired signalling to arrest and repair, and their tissues respond badly even to modest doses. When clinicians began using radiotherapy in the early 20th century, and through the mid-century's expansion of oncologic practice, They learned empirically that some tissues tolerate radiation better than others. Modern molecular biology explains why, and the choices of individual cells cascade up. When a critical mass of stem cells is lost, whether in bone marrow niches or in intestinal crypts, the tissue's regenerative logistics fail. The stem cell is not merely a replicator, it is a member of a community, the niche composed of stromal cells, extracellular matrix, blood vessels, and signaling molecules. Radiation does not spare the niche. Stromal cells become senescent, blood vessels leak, and the supportive environment that fosters stem cell self-renewal collapses. Without that microenvironment, even surviving stem cells may be ineffectual. Pioneers of bone marrow arms transplantation, most notably E. Donald Thomas in the United States, began in the 1950s and 60s to address marrow failure by providing new stem cells and a new microenvironmental template. Transplantation became a tool not only in treating hematologic malignancies, but in responding to marrow aplasia from radiation exposure. The logistics are daunting: matching donors, conditioning regimens, infection control. The scale of a mass casualty event can quickly exceed the capacity of transplantation as a response. Minute events become tissue narratives. Within minutes to days after a radiation insult, endothelial cells lining the microvasculature respond. Endothelium is exquisitely sensitive to oxidative stress. It responds by expressing adhesion molecules, increasing permeability, and, at higher doses, undergoing apoptosis. The leakiness allows plasma proteins and inflammatory cells to seep into tissues, the first phase of a remodeling program that, over weeks and months, can become fibrosis. Vascular damage also promotes thrombosis and chronic ischemia. The lung provides a canonical example. Early endothelial and epithelial injury after thoracic irradiation can lead to pneumonitis in weeks to months, characterized by cough, dyspnea, and a storm of inflammatory cytokines. Later, in the months to years, the tissue remodels into fibrotic scarring that impairs gas exchange permanently. Those late effects were documented in patients receiving mediastinal radiation for Hodgkin lymphoma in the mid-20th century. As cohorts aged, epidemiologists in Europe and North America detected increased rates of cardiac disease and pulmonary fibrosis in survivors whose therapy had been state-of-the-art in the 1960s and 1970s. The gut is an unforgiving example of how microlesions translate swiftly into organ-level failure. The intestinal epithelium is a conveyor belt of cells produced by crypt stem cells and pushed outward. Radiation kills the crypt progenitors. In the hours after exposure, apoptosis spikes in the crypt base. Within days, the lining sloughs, villi collapse, and the protective barrier is breached. Bacteria, previously contained within the lumen, find the bloodstream. Infections, sepsis, and profound fluid loss ensue. The clinical picture is not only a local failure, it is a logistic collapse, lost absorptive surface, electrolyte derangement, and an overwhelmed immunity because the bone marrow is also likely wounded. The combination is lethal at higher doses. Surgeons and physicians in the years after atomic testing and nuclear accidents learned, sometimes in grim fashion, that certain organ systems are early responders to whole-body exposure, and that clinical triage must prioritize supportive measures for the marrow and the gut. Skin is another front. It is visible, and thus often the focus of public concern after accidents, but its role is both sentinel and structural. Radiation dermatitis evolves from an acute erythema, a sudden redness like a burn, to chronic ulceration, and sometimes necrosis if deep tissue damage interrupts blood supply and regeneration. Historical accounts from the radium girls in the 1920s through the earliest X-ray technicians who did not appreciate dose precautions to patients undergoing radiotherapy all taught the medical community about fractionation. Smaller daily doses give normal tissues recovery time while still damaging tumour cells. This logistical insight, refined by radiobiologists in the mid-20th century, is a strategy that harnesses fate decisions at cellular level and applies them at scale to salvage function while treating disease. Repair, failure, hergia, and regeneration are conversations across time. After the initial storm of cytokines and cell loss, tissues either organize to repair or they brazenly remodel into a new homeostatic state that may not resemble the original. The liver, blessed with a remarkable regenerative program, often recovers from substantial loss, provided its vascular scaffold and stromal support remain. The heart and brain are less forgiving, Loss of cardiomyocytes or neurons tends toward permanent functional deficit. In such organs, vascular injury and glial or fibroblast activation lay down scar, a substitute that cannot contract or compute. Those later outcomes, heart failure years after thoracic radiation, cognitive decline after cranial irradiation, were observed as radiotherapy matured as a cancer treatment and as survivors aged into the late effects that had been invisible in the era when most patients did not survive long enough to reveal them. They taught clinicians to think as logisticians, mapping not only immediate efficacy but long-term functional cost. Scale matters. A single irradiated cell in the middle of a lung lobe is a nuisance. 1,000 coordinated failures shift an organ's function. In the aftermath of Chernobyl, the acute deaths were concentrated among plant staff and firefighters who absorbed very high doses in a short time. But the long tail of cancer risk spread across millions in Belarus, Ukraine and Europe because of the logistic reality of radioactive iodine dispersion, dietary habits, and the age structure of populations. Children swallowed iodine 1v1 concentrated in milk. Their thyroids avidly accumulated the isotope and bore the molecular signatures to rearrangements that became thyroid cancers years later. Public health responses that distributed potassium iodide quickly could have mitigated much of that risk, but the logistics on the ground in April 1986 were chaotic. Warnings came late, supplies were limited, and the scale of displaced populations complicated an organised prophylactic campaign. History in these cases is not a corpus of anecdotes. It is a ledger of logistic successes and failures that changed who developed disease and who did not. At organ level, 3 processes determine functional outcome: stem cell depletion, stromal alteration, and impaired regeneration. Stem cells are the future of a tissue. Without them, no recovery is possible. Stromal cells, fibroblasts, endothelial cells, immune components, create the conditions for stem cell survival and differentiation. Radiation alters stroma in ways subtle and persistent. Fibroblasts can become activated into myofibroblasts that deposit collagen and seal the tissue in scar. Endothelial loss reduces capillary density and creates hypoxia. Immune cells, initially recruited for cleanup, may linger and secrete factors that prevent constructive regeneration. The interplay is a closed loop of pathology. In bone marrow, for instance, stromal damage means that even transplanted hematopoietic stem cells may have trouble engrafting if the niche is hostile. In chronically irradiated muscle or skin, persistent hypoxia and a fibrotic matrix make mechanical function hold up difficult. Surgeons and rehabilitation specialists, looking at victims of radiologic exposures and at cancer survivors in clinics across Boston, Tokyo, Kiev and elsewhere, have learned these patterns through decades of gradual observation. What tissues can be coaxed to renew, and what tissues must be compensated for through prosthetics and supportive care. Clinically, that translates into predictable syndromes and logistical imperatives. Bone marrow failure demands blood products, antibiotics, and growth factors. At higher severity, it requires transplantation, a resource-intensive multidisciplinary intervention that calls for infrastructure, cold chain logistics, matching registries, and long-term follow-up. The gut requires fluid resuscitation, nutritional support, and sterility when possible, but the loss of mucosal barrier invites a cascade of infections that even the best antibiotics may not contain. Pulmonary and cardiac late effects require chronic care models, pulmonary rehabilitation, and cardiology follow-up that health systems must plan for decades in advance. The historical lessons of large-scale exposures, Hiroshima and Nagasaki after 1945, the atmospheric tests of the 1950s and 60s that altered diets and risk in populations, Chernobyl in 1986, the troubled reactor at Fukushima in 2011, are as much about medical triage and long-term health systems planning as they are about immediate pathology. The science of DNA damage and repair sits at the smallest scale, but the consequences demand the largest logistics. There is a final, quiet irony to the process. Cells that deploy apoptosis, that choose to extinguish themselves cleanly, often spare their neighbours a fiery inflammatory aftertaste. Yet an organism cannot simply cull endlessly, it needs its tissues. A population of cells that stubbornly survives with chromosomal rearrangements, however, can occupy the tissue for years, quietly dividing, slowly accruing further mutations or epigenetic changes that permit invasion, angiogenesis, and metastasis. That is the long-term cost of misrepair, a slow mutation-driven tail, that can culminate in cancer decades after an initial insult. The timeline in human history is long. The sequelae of exposures in the 1940s and 1950s continue to inform policy. The consequences of Chernobyl were still being mapped in the 1990s and 2000s, and the concerns about low-dose exposures and genetic risk persist in risk assessments today. Researchers and clinicians have become, in effect, archivists of damage, tracing mutational signatures back to likely exposures and piecing together the social and environmental logistics that created the risk. So the story moves from the immediate, a strand sliced, an enzyme racing to bind, through cellular fate decisions made in hours and days, to tissue-level responses that unfold over weeks, months, and years. Molecules misjoined in a sudden burst can create a rearrangement that by accident and Darwinian selection becomes the foundation of a new clonal dynasty. Cells that choose senescence alter their neighbourhood with inflammatory whispers. Cells that die create gaps the body must fill. Cells that survive with damage seed future disease. The scale of the event, a laboratory accident in one hospital ward, the deliberate detonation of a weapon in a city, the failure of a reactor cooling system on a spring night, changes the logistics of response, the number of tissues involved, and the public health strategies required for mitigation. The biology, however, is constant. DNA is a baton passed from one generation of cells to the next. And when it is miswritten or misstitched, the echoes of that failure roll forward in time, shaping who recovers, who suffers late consequences, and who years hence finds cancer in a tissue that once seemed whole. In the end, the tale is not merely one of molecular mechanics, but of choices at every scale. Cells choose to die or to persist. Clinicians choose how to triage and whom to treat. Societies choose how many resources to devote to prevention, to shelter, to stocking potassium iodide, and to long-term surveillance. The mechanisms, point mutations, deletions, translocations, micronuclei chromothripsis, breakage fusion bridge cycles, are the grammar of a story that can be told in gene sequences. The fates, apoptosis, senescence, autophagy, necrosis, mitotic catastrophe, are the verbs that describe the immediate outcomes. The tissues and organs, with their vascular scaffolding and stem cell niches, are the settings where the drama plays out, often slowly, often with a stubbornness that demands decades of care. History informs us that this interplay is not merely theoretical. It has practical impact on maladies, on medical systems, and on policy. The sombre fact, sometimes delivered dryly in the pages of a report, is that a single misrepaired break, a single misjoined chromosome can ripple outward, and in time, with the patience of evolution and the indifference of cells, can change lives long after the initial light and heat have faded. The instant a photon or particulate track slices through human tissue, it feels, at the molecular scale, like a sudden winter lightning. No sound for us, only a cascade of invisible charges and chemistry that unthreads the familiar order of molecules. In the first trillionths of a second, electrons are stripped, bonds are broken, and for a breath the cell's interior becomes a storm. Water, which makes up nearly 3/4 of our soft mass, is the first to pay. The tracks of ionization produce hydroxyl radicals, hydrated electrons, hydrogen atoms, tiny chemical assassins that live for microseconds but rewrite fates. These radicals nick sugars, alter bases, and most consequentially, if you are a cell that remembers its heredity, create single-strand breaks and double-strand breaks in DNA. A convenient rule of thumb, learned in laboratories from the 20th century onward, is that a whole-body dose of about 1 gray will generate on the order of 1,000 single-strand breaks and roughly 40 double-strand breaks per human diploid cell. The numbers are cold arithmetic, but their effects are anything but. Those 40 double-strand breaks per cell are the locus of carcinogenesis, of instant cellular death, of lineage derailment. A single unrepaired double-strand break can be repaired faithfully, misrepaired into a translocation, or left to fester and be converted to a mutation at the next replication. Within seconds to minutes, molecular sentinels sense the damage. ATM and ATR kinases ripple signals across chromatin. P53 wakes from its steady vigil and decides, in part by numbers, whether the cell will pause and repair or trigger programmed death. When a small fraction of cells in a tissue carry a manageable burden, repair systems and cellular redundancy carry the organism through. When a large fraction is hit, when a significant proportion of stem cells in marrow or crypt cells in the gut are damaged, the architecture of tissue collapses, and the timeline of symptoms accelerates from days into hours. That acceleration is the essence of acute radiation syndromes and the reason we speak of dose-dependent tiers, hematopoietic, gastrointestinal, and neurovascular, each mapped to a progressively higher range of absorbed dose and each unspooling over characteristic hours to weeks timelines. At low but clinically meaningful whole-body doses, roughly on the order of 1 gray, the story reads as a slow betrayal. Minutes to hours after exposure there is a prodrome, nausea, a wave of weakness, sometimes vomiting. These are not yet the life-threatening symptoms, they are the body's first telegram that something has happened. For survivors of Hiroshima and Nagasaki, medical teams and later the Atomic Bomb Casualty Commission recorded many such prodromal episodes in 1945 and the years that followed. In modern language, these early signs signify systemic signalling, intrachrome affin cells releasing serotonin after damage to intestinal epithelium, vagal stimulation, and cytokine surges that make a person feel unwell, while the real cellular accounting proceeds quietly. Days to weeks later, the hematopoietic syndrome appears, because marrow is a tissue of constant turnover and its stem cell pool is limited. When a sufficient fraction of hematopoietic stem and progenitor cells are killed, when their clonogenic capacity is lost, the blood cannot replenish itself. White cells fall, platelets drop, anemia deepens. The calendar of decline is slow enough to be deceptive. A latent period that can feel like recovery stretches for days as peripheral counts slowly sink. Then infection, hemorrhage, and anemia impose themselves. Without antibiotics, transfusions, or stem cell rescue, doses in the range commonly cited as the human LD50-60, roughly 3 to 4 grays in the mid-20th century medical context, translated into a 50% mortality within 60 days. That number was not merely laboratory trivia. It became a planning metric for hospitals and civil defense agencies during the Cold War, a grim piece of arithmetic informing triage and the allocation of scarce resources. Later, as antibiotics, parenteral nutrition, blood banking and bone marrow transplantation advanced, the LD50 shifted upward, a small mercy bought by decades of medical development. Past roughly 6 grays, the intestinal tract becomes the brittle hinge on which survival turns. The gastrointestinal syndrome is not simply diarrhea and dehydration. It reflects the near-total eradication of the crypt stem cells that renew the mucosal surface. Within hours to days, the prodrome is severe. Within days, the mucosa sloughs, naive bacteria flood the lamina, and fluid losses and translocation of gut flora set up sepsis. The cause of death in this band of dose is multifactorial. Fluid and electrolyte derangement, cascading infection, and the systemic inflammation that follows massive epithelial loss. Time compresses, where hematopoietic failure unfolds over weeks. Gastrointestinal collapse kills within days to 2 weeks. This is precisely why planners who have studied nuclear accidents and tests speak so often of the window for medical rescue, a narrow, unforgiving interval in which reconstitution therapies must be delivered, logistics marshalled and supply chains kept open. Above 2 to 3 dozen grays, or at doses sufficient to cause massive capillary and endothelial failure in the central nervous system, the neurovascular or central nervous system syndrome announces itself in cruelly brief time. Progressing within hours, sometimes minutes, the clinical tableau is driven less by DNA damage to post-mitotic neurons and more by radiation-induced endothelial dysfunction and blood-brain barrier breakdown, cerebral edema, increased intracranial pressure, loss of autonomic control, convulsions, coma. These are rapid, overwhelming endgames. The cellular accounting here is immediate. Endothelial cells, which maintain microvascular integrity, lose function, and the brain, dependent on an exquisitely controlled environment, succumbs quickly. At Chernobyl in April 1986, the initial victims, firefighters and plant workers who received massive localized exposures, developed precisely this pattern. Hospital registries documented rapid neurological deterioration alongside classical hematopoietic and gastrointestinal injuries. The Soviet clinicians who recorded those early cases learned, under impossible pressure, the same lesson as researchers in Hiroshima. Higher dose begets shorter time. Across these bands, the prodrome and latent periods are not merely calendar entries, but reflection of cellular calculus. Seconds matter because each ionization is a potential double-strand break. Minutes matter because the balance between repair and misrepair is being decided. Hours and days matter because whether stem cells die or survive determines whether tissues can regenerate. At a mechanistic level, high doses precipitate rapid systemic failure because they destroy not only parenchymal cells but the support systems, marrow stem cells that make the immune system, crypt stem cells that maintain nascent barriers, endothelial cells that keep blood and tissue apart. The subsequent cascade, bacterial translocation, hemorrhage, sepsis, disseminated coagulopathy, multi-organ dysfunction, is an ecological collapse inside the body, a loss of the small, specialized populations that sustain life's basic transactions. Where those populations are abundant and replaced rapidly, damage appears sooner. Where cells divide slowly, damage may smoulder and show itself later, or not at all in the acute phase, but the story does not stop with collapse. The same first seconds that produce catastrophic cell death in some cells also leave other cells alive but scarred, carrying molecular wounds that will be written into their progeny if given the chance. Those misrepaired lesions, chromosomal translocations, deletions, point mutations, are the seeds of the long tail of carcinogenesis. The historical record supplies the evidence. Roef's decades of follow-up of atomic bomb survivors in Hiroshima and Nagasaki showed an early spike in leukemia beginning about 2 to 3 years after exposure and peaking within the first decade, followed by a steady excess of solid tumours that unfolded over decades and continues to inform risk models to this day. Acute leukemias are often the fastest clinical manifestation of radiation's mutagenic power because hematopoietic cells divide quickly and because a single driver mutation in a stem cell can, under the right selective pressures, clonally expand to dominate a lineage within years. To understand why a single double-strand break in a stem cell can be the first domino in a cancer that appears years later requires walking across scales of biology. Immediately after exposure, the damage is chemical and structural. DNA is gapped, bases oxidized, chromosomes fractured. Repair pathways deploy, non-homologous end joining is fast but error-prone, and homologous recombination is more accurate but requires a sister chromatid and thus occurs during SG2 phases. If a break is misjoined, a fusion between chromosomes can occur, producing dicentrics or translocations. If a repair leaves a deletion, tumour suppressor genes may be lost. If a point mutation creates a constitutively active kinase, growth control is altered. But a mutation is not a cancer. For malignancy to emerge, the mutated cell must survive immune surveillance, outcompete neighbours, and navigate microenvironmental selection pressures that favour expansion. This is why tissue turnover matters. In epithelia with rapid renewal, a mutated progenitor that retains clonogenicity can expand more quickly than in non-renewing tissues, so cancers of the gut and blood show shorter latencies than those of the brain or muscle. The stochastic nature of carcinogenesis, that the probability, not the severity of cancer increases with dose at low to moderate exposures, makes the phenomenon both maddeningly unpredictable and statistically tractable. For any single person exposed to a low dose, the outcome is a roll of biological dice. Across populations, the rolls average out. Epidemiology, therefore, is the historian's microscope for cancer from radiation. The ABCC and later the RERF cohorts provided the long-term data that allowed committees such as BIR, Biological Effects of Ionizing Radiation, and UMSC-ER to construct dose-response models and give public health agencies numbers to guide regulation. In the early 21st century, the BR7 report endorsed, for low linear energy transfer radiation, a linear no-threshold model for cancer risk as a pragmatic basis for radiation protection. That model is not without critics. Physiologists and radiobiologists continue to study dose rate effects, thresholds for specific endpoints, and the possibility of hormetic responses at vanishingly low doses. The physics is clean, the biology is probabilistic. Policy reflects the compromise between them. At the molecular level, the path from lesion to fixed mutation often demands replication machinery. A DNA adduct or break that is accurately repaired before S phase leaves no legacy. If the cell is pushed to replicate with imperfect reprisal, the lesion is fixed as a mutation. This is why cell cycle checkpoints matter as guardians of genomic truth. p53-mediated arrest allows time for repair. Failure to arrest, whether through p53 mutation or overwhelming damage, permits error to be consolidated. For stem cells, which must self-renew across years, a fixed mutation is especially consequential. In bone marrow, an HSC that survives radiation with a driver mutation can undergo clonal expansion, sometimes detectable as clonal hematopoiesis decades before any frank leukemia, transforming a single second's damage into a long-term altered state. In solid organs, a mutated epithelial stem cell lurks in tissue microenvironments shaped by aging, inflammation, and occasional further mutagenic hits. These pressures select for expansions that become the microscopic foci of eventual tumours. The signature of radiation in cancers is not always unique in terms of single nucleotide changes, but it often reveals itself through structural complexity, complex chromosomal rearrangements, loss of heterozygosity, and deletions that betray error-prone repair of double-strand breaks. In therapy-related leukemias, observed after high-dose radiotherapy or cytotoxic chemotherapy, characteristic cytogenetic patterns, chromosome 5 and 7 deletions for example, have been identified, connecting clinical experience to molecular mechanism. Those examples served as warnings to clinicians and shaped protocols for bone marrow transplantation and follow-up surveillance. The same theme recurs in epidemiology. Leukemias cluster earlier than epithelial cancers. Thyroid cancers rose markedly in children after Chernobyl's Iowa-31 fallout, with latency measured in a few years rather than decades. Breast and lung cancers among atomic bomb survivors showed rising excesses decades later. The time between the initial ionization and clinical cancer is a ledger of tissue biology, repair dynamics, and selective environment. This ledger extends into the realm of reproduction and development, where timing is everything. The mid-20th century geneticists who first mapped radiation's germline effects set the conceptual map. Hermann J. Muller, his Drosophila work in the 1920s and 1930s, culminating in a Nobel Prize in 1946, showed clearly that X-rays increase heritable mutation rates in fruit flies. In the years after 1945, mammalian work, William L. Russell's specific locus tests in mice among them, quantified that gametes exposed to ionizing radiation acquire mutations at elevated rates. Those animal studies established the principle that radiation is a potent germline mutagen. They also allowed researchers, through carefully timed irradiation across stages of gametogenesis or embryogenesis, to map windows of vulnerability. Spermatogenesis is a multi-week process in mammals. A stem spermatogonium damaged today can produce defective sperm weeks later. Oogenesis is different. Human oocytes are formed in the fetal ovary and arrested. Damage to stored oocytes can deplete the reserve, causing infertility or early menopause years on. The practical upshot is asymmetric sensitivity. Male preconception exposures tend to alter the risk landscape across to future conceptions in a way that is time-dependent and linked to spermatogenic waves. Female exposures can deplete or damage finite ovarian reserves, or, if they occur during pregnancy, directly affect the developing embryo. Human epidemiology has been less dramatic than the animal data in one particular sense. The RERF analyses of children born to atomic bomb survivors have not shown a clear, large increase in heritable genetic disease compared to expected baselines. That absence of a clear signal is not a refutation of the biological effects seen in laboratory systems. Rather, it reflects statistical realities, the modest sizes of measurable effects when distributed across human population variability, and the strength of repair and selection during reproduction. Still, human pregnancy is exquisitely sensitive at certain narrow windows, and the Hiroshima, Nagasaki in utero data taught clinicians and scientists a lesson that laboratories had predicted. Embryos exposed in the pre-implantation window, the earliest 2 weeks, follow an all-or-none rule. Radiation tends to either kill the entire embryo or leave a survivor that develops normally, because the early embryo is a small set of cells whose loss usually aborts the pregnancy. During organogenesis, roughly weeks 2 through 8, radiation can produce malformations by killing lineage-committed progenitors. If enough cells in a forming organ die, the architecture cannot be completed, and a structural defect results. The central nervous system, however, is vulnerable later. RIAF's careful cohort analyses showed that exposures in weeks roughly 8 to 15 of human gestation correlated with increased risks of microcephaly and severe intellectual disability. Those observations have precise, somber meaning. At those gestational weeks, neuronal proliferation and organization are at a peak. Radiation-induced loss of neural progenitors translates into fewer neurons and disrupted circuitry, effects that manifest years later as cognitive impairment. Practical human responses to these vulnerabilities have been historical and logistical, as much as medical. After Chernobyl, the greatest public health success, and the bitter lesson, was about iodine prophylaxis. Radioactive iodine concentrates in the thyroid, especially in children who drink contaminated milk. In Belarus, Ukraine, and parts of Russia, delayed evacuation and uncontrolled milk distribution meant many children received substantial iodine doses. Within a few years, clinicians saw a sharp rise in childhood thyroid cancers. By contrast, in the immediate aftermath of Fukushima in 2011, lessons learned from Chernobyl and decades of public health planning led to rapid distribution of potassium iodide and prompt evacuation of at-risk populations, actions that substantially mitigated thyroid doses in children and likely reduced the magnitude of any future thyroid cancer excess. The Marshall Islands experiences after the 1954 Castle Bravo test, Rongelap and other atolls contaminated with fallout, are another historical chapter in which evacuation and monitoring were the primary public health responses, followed by decades of epidemiologic follow-up to understand long-term outcomes. When we consider reproductive risk, we must also contend with another truth. The germline's exposure does not produce simple, predictable outcomes like a broken bone. Mutations induced in sperm oocyte DNA may be eliminated by early embryonic selection, may be compensated by maternal transcripts and repair mechanisms at fertilization, or may be passed to offspring and interact with myriad other genes and environments across lifetimes. Consequently, while animal experiments demonstrate clear heritable mutation effects and inform precautionary policy, human genetic consequences at population scale have been subtle, masked by the vast background of spontaneous mutations and selection. That scientific nuance is as often lost in public conversation about radiation and babies, which is one reason historical literacy matters. The data collected from Hiroshima and Nagasaki, from Marshallese medical histories, from Chernobyl registries, and from controlled animal experiments together, create a graded picture that policymakers translate into practical counsel, screening, counselling, and, when appropriate, evacuation and prophylaxis. A final important thread in this long tapestry is the role of dose rate and quality. A grey delivered as a single intense flash is biologically different from the same grey spread over weeks, months, or years. The intervening time allows repair systems to act and immune surveillance to clear damaged cells before they clench into malignancy. Similarly, the type of radiation matters. Alpha particles, with high linear energy transfer, make dense ionization tracks that are especially effective at producing clustered DNA damage and complex chromosomal breaks. That is why internal emitters, radioisotopes that lodge in bone or thyroid, can produce organ-specific risk profiles very different from whole-body low-LET exposures. This interplay of rate, quality, and tissue specificity has been unravelled piece by patient piece across the history of radiobiology, from the Drosophila cages of Muller to the epidemiological cohorts that continue to be followed in Japan, Belarus, Ukraine, and elsewhere. If there's a sombre, almost personal moral to the historian's telling, it is that radiation is both immediate and long-lived. An event that lasts seconds can shape decades of biology. The initial storm of free radicals and strand breaks is quick. The clinical consequences unfurl on multiple clocks, some alarms ringing within hours, others years later as cancers, and in some cases quiet changes in the germline that never fully announce themselves in the human record. Scientists, clinicians, and public health officials have learned to read these clocks. They have built institutions, ABCC, later ref, UNSCR, BIR, and National Radio Protection Agencies, that translate seconds into policies and decades into surveillance. They have learned to ration antibiotics and blood when a city is overwhelmed, to distribute iodide and evacuate children when fallout falls, to monitor cohorts for subtle increases in cancer and reproductive outcomes, acknowledging that the human sample sizes required to see small effects are large, and that careful follow-up is a moral requirement. So the narrative returns quietly to the cell. Within that first invisible flash, a cell either dies, repairs, misrepairs, or survives carrying a new code. It is a decision made at molecular speed, but with lifetime consequence. Across thousands and millions of such microscopic choices, population-level risk becomes an arithmetic we can model, predict, and to a degree mitigate. The arc of history, Muller's bench, Russell's mice, the doctors in Hiroshima and Nagasaki, the physicians in Pripyat, the public health teams in Sapporo after 2011, reads like a cumulative experiment in understanding radiation's dual temporality, brutal immediacy and patient latency. And in that folded time, as the sleepless historian would whisper, there is also a human lesson about preparedness, that the speed of the physical insult and the slowness of some of its consequences demand both rapid action and decades of attention. Time, to radiation, is a measure of consequence. Time for us must be a measure of responsibility. Measuring exposure begins with a language for the invisible, the grey and the civet, siblings born of the need to translate energetic trespass into numbers that a surgeon, a regulator, a worried parent can grasp. The grey symbol G is simple and stern. 1 joule of energy deposited per kilogram of matter. If a beam of X-rays or the whoosh of a slow drip of a radioactive isotope gives tissue its heat-like burden, the grey counts the energy that was actually absorbed. The civet SV is a different sort of accounting. It is the grey wearing a tailored suit of context. It multiplies absorbed energy by factors that try to capture the biological whisper behind raw physics. Where grey tells you how hard the invisible bird has hit you, civet asks what kind of feathers the bird carried. Alpha, beta, gamma, which organ was struck, and therefore how much mischief might follow. Older names still hang in the hallways. The Röntgen that measured ionization in air, the rad and rem that once dominated labs and clinics, handoffs in a technical language that matured through the 20th century till the SI systems settled on grey and civet in the international conversations of the 1970s. Those two measures are the starting point, but the story of damage is a story of timing. An absorbed joule spread thinly over a year is not the same as the same joule detonated in a single breath. Dose rate, the flow of energy through time, is a governor on biology. A pulse that arrives all at once, as in the microsecond scream of a nuclear detonation, saturates repair crews and kills without warning. Slow, protracted exposure invites repair, adaptation, and sometimes compensation. For radiobiologists and clinicians, this is the law. The same total absorbed dose can be more or less harmful depending on how hastily it was given. That simple observation is the bedrock of fractionation in radiotherapy, the reason why cancer treatments are parcelled into days and weeks rather than delivered as a single brutal blast, and the reason why the life of dial painter wiping radium on watch faces over years is not the same as a miner standing a day near a leaking source. If the grey and civet are the units of law, then linear energy transfer and relative biological effectiveness are the character sketches. Linear energy transfer measures how concentrated those joules are along a hairline path, usually expressed in keV per micrometer. A heavy charged particle like an alpha leaves a dense scorched trail of ionizations and chemical havoc in its wake. High LET. A photon, a gamma ray, here more often scatters and sprinkles its energy broadly. Low LET. Biologically, that makes all the difference. High LET radiation produces complex clusters of molecular lesions, many of them too knotted for routine repair machinery to untangle. The cell sees damage that reads like a ransom note rather than a scribble. Relative biological effectiveness, RBE, is the shorthand for the comparison. How much more or less lethal a given form of radiation is compared to a reference, usually 250 kVp X-rays or cobalt-60 gamma rays. RBE is not a fixed number. It is shaped by LET, by dose, by the kind of tissue, and by the crucial dimension of time. The same alpha track that causes near-instant cell death in one context might yield mutation without death in another. In that small world between molecule and cell lies microdosimetry, the discipline that bridges physics and biology by asking how energy lands at microscopic scales. Macrodosimetry can tell a department head that an irradiator deposited half a grey into a patient's chest. Microdosimetry asks how many of those joules found themselves in the nucleus of a single stem cell, how many clustered within a few nanometers of DNA, and whether the incised damage was likely to be sloppy enough to produce misrepair and mutation. Track structure models, Monte Carlo simulations and microbeam facilities have turned what was once a poetic metaphor, radiation scouring, into quantifiable landscapes. They show us again and again that a single alpha particle threading a nucleus can create a constellation of double-strand breaks, base damage and crosslinks that are difficult to piece back together faithfully. Low-let photons may create more single-strand breaks spread across hundreds of cells. The cell's repair teams, in such a case, have a chance. High-let insult gives repair no breathing room. The practical consequence is immediate. For internal emitters like polonium or plutonium, which deliver localized high-let energy to small volumes of bone or lung, a modest number of becquerels can produce effects far out of proportion to their nominal absorbed dose. Time is uncompromising. In the first instance after a photon collides with tissue, within femtoseconds, electrons are liberated and molecular bonds are ruptured. In the next billionths to microseconds, free radicals form and begin a chemistry of oxidation that will persist long after the electromagnetic wave has passed. Within microseconds to seconds, DNA strand breaks appear. Within minutes and hours, sensors and scaffold proteins like ATM, ATR, Pu53, and an orchestra of kinases marshal checkpoints, stop cell cycles, and commence repair. Some damage is rejoined neatly, some is misjoined, producing small insertions, deletions, or chromosomal translocations. Some cells, sensing the extent of injury, die by apoptosis to spare the tissue a corrupted offspring. Those decisions, repair, misrepair, senescence, apoptosis, occur on a canvas of hours and days. Yet the consequences of misrepair may be printed into genomes and live for decades as cancer. What matters, therefore, is not simply how many joules were absorbed, but the precise choreography of their delivery and the immediate cellular response. This hard clinical appreciation of physics and time was not conjured from first principles. It was hammered into place by history's unquiet experiments, by failures and by the stubborn diligence of observers who kept count. Wilhelm Röntgen's dim lab in Würzburg in November 1892 yielded a glow on a nearby screen, and with it a revolution. Within months, doctors in Berlin, Paris and New York were illuminating bones and treating tumours with X-rays. Within the year, skin burns were reported. Precaution lagged behind possibility. In the United States, one of the earliest, saddest, full-length cautionary tales involved Thomas Edison and his assistant Clarence Daly, who in the turn of the century handled experimental X-ray tubes until the tissue of his arms and hands ulcerated and became malignant. Daly died in 1904. Thomas Edison, pragmatic and never sentimental about commerce, famously gave up X-rays not wholesale because he was cruel, but because the cost in his laboratory tasted too bitter. Others pressed on, and the early 20th century saw both therapeutic miracles and avoidable harm. Radioactivity, discovered by Henri Becquerel in 1896 and rendered more tangible by Marie and Pierre Curie in 1898 with the isolation of radium, carried its own set of tragedies. Radium shimmered in the laboratory and in the marketplace. It was sold as a vitalizing powder and used in everything from watch dials to health tonics. The young women who painted luminous numbers on watch and instrument faces, the radium girls in factories in Orange, New Jersey and Ottawa, Illinois, learned a lesson the market refused to teach, that ingesting tiny beads of radium by lips and breath deposits high at alpha emitters in jawbone and marrow, and the body pays with necrosis, anemia and carcinoma. Grace Fryer and other plaintiffs brought their case to court in 1927, and although the legal battles stretched, the public attention forced factories to change practices and regulators to take notice. The images from those courtrooms, skeletal X-rays showing starved bone, glowing evidence on photographic plates, shaped a public health awakening. It was not a clean or quick correction, it was iterative and painfully human. If small industrial tragedies sowed the seeds of occupational protection, nothing in the 20th century concentrated public and scientific attention like the atom. Hiroshima and Nagasaki, August 6th and 9th, 1945, were instantaneous translations of energy into human fate. Survivors close to the hypercenters experienced doses measured in grays in literal seconds, flash burns, immediate cell death in the gut and marrow, and a later bloom of leukemia that appeared within a few years. The United States and Japan established the Atomic Bomb Casualty Commission in 1947 to follow survivors and to record what an instantaneous enormous dose did over a lifetime. The lifespan study that grew from that cooperation, later conducted by the Radiation Effects Research Foundation, provided the statistical backbone for our models of stochastic effects, for risk projection over decades, and for the unsettling lesson that even single early events can leave signatures that persist across generations of cells and people. Radiation epidemiology learned from these cohorts that some effects, like leukemia, can have relatively short latencies after high doses, while solid cancers often take decades to manifest. Both bore dose-response relationships that scientists would spend generations mapping and arguing over. Clinical radiotherapy occupies an ambivalent place in this history, a therapy born of the same physics as harm, refined over decades into a precise art that both kills and cures. In the Paris laboratories and clinics of the 1920s and 30s, people like Henri Coutard experimented with fractionation, the idea that dividing a total dose into many small exposures allowed normal tissues to recover better than tumours, improving the therapeutic ratio. These were empirical observations at first. Later, Louis Harold Gray and his colleagues in England in the mid-20th century brought more systematic measurement to questions of oxygen effect, cell survival curves and fractionation biology. Hermann J. Muller's work in the 1920s with Drosophila, showing that X-rays induced heritable mutations, carried political as well as scientific weight and fed into debates about genetic risk. In laboratory benches across Europe and America, methods matured. The Geiger-Müller tube became a staple of detection in 1928. Film badges and later thermoluminescent dosimeters offered people the modest armour of knowledge about how much exposure they carried, and the clonogenic assays of Puck and Marcus in 1956 gave radiobiology a quantitative handle on surviving fractions of irradiated cells. Those experimental forms, in vivo experiments, colony formation assays, track structure calculations, converged on models like the linear quadratic formalism that clinicians would use to plan dose and schedule. Not every formative episode was clinical or accidental in the industrial sense. Many were laboratory contrivances that revealed the subtler features of damage. The discovery in the 1990s of the radiation bystander effect, that non-irradiated cells adjacent to irradiated ones sometimes show DNA damage and stress responses, complicated the simple picture of hit cells and untouched neighbours. Single-cell microbeam experiments, where scientists could aim a particle at the nucleus of a single cell and watch the cascade of molecular events, offered visceral clarity on how clustered lesions could defeat repair. The realization that some molecules, most famously the phosphorylated histone GH2X, appear at double-strand break sites within minutes gave clinicians a direct, though ephemeral, marker of damage. Practical medicine folded these scientific truths into policy slowly. Regulators created exposure limits. International bodies like the International Commission on Radiological Protection, ICRP, founded in 1928 and reshaped over subsequent decades, translated experimental and epidemiologic data into guidance on how to balance utility and risk. Then there were the spectacles of system failure that taught the world about both instantaneous insult and slow lingering contamination. The Soviet reactor accident at Chernobyl on April 26, 1986 mixed a local catastrophe of explosions and fires with a geopolitical fog of secrecy. Immediate fatalities and acute radiation sickness among firefighters and plant staff were visible and brutal, but the true scale of human cost unfurled over years. Iodine-131 in milk and food chains seeded a wave of thyroid cancers among children in Belarus, Ukraine and parts of Russia. Cesium-137 lingered on fields. Whole villages were labelled uninhabitable. The world learned about the logistics of evacuation, the politics of warning populations, and the maddening delays that secrecy can inflict on triage. Nearly a quarter of a century later, the Fukushima Daiichi accident of March 11, 2011 offered a different set of lessons: the interplay of evacuation strategy, the distributions of contamination, the role of potassium iodide prophylaxis when iodine isotopes are present, and the psychological, social and economic consequences that outlast measurable doses. Each episode etched on the scientific memory the same injunction: the pattern of exposure, a single surge, a prolonged drizzle, an internalized source, shapes biological outcomes, social choices, and regulatory responses. Out of these episodes came policy, medicine, and some practical things that can be reached for in an emergency. Regulators learned to build guardrails, the concept of maximum permissible dose for workers, the distinction between stochastic effects that grow in probability with dose and deterministic effects that occur above thresholds. and the practical rule of time, distance, and shielding. Triage protocols for acute radiation syndrome exploit seconds-by-second phenomenology. The timing of nausea and vomiting after exposure, the precipitous fall in lymphocyte counts, early biomarkers such as the kinetics of neutrophil loss, these have been correlated empirically with dose ranges and are used to prioritise care when numbers are large and resources finite. Stockpiles of potassium iodide can blunt thyroid uptake of radioactive iodine if distributed quickly. Chelation strategies using Prussian blue for cesium decorporation or DTPA for certain actinides can reduce body burden when internal contamination occurs. Supportive medicine has also advanced. Granulocyte colony stimulating factors like filgrastim, now part of many national emergency caches, can accelerate marrow recovery. Antibiotics, transfusions, and in extreme cases, hematopoietic stem cell transplantation are components of the clinical playbook. Those interventions do not erase the seconds of the initial assault, but they can change the trajectory from fatal collapse to recovery and reduce late complications. In clinical oncology, the seconds and their distribution have been weaponized with increasing finesse. Modern radiotherapy is a study in temporal and spatial control. Stereotactic radiosurgery and stereotactic body radiation therapy compress high doses into very small targets. Proton and heavy ion therapy exploit the Bragg peak, so that energy is deposited at a designed depth rather than sprayed over all tissue. For high-latency heavy ions such as carbon, the RBE is higher, and that can be a therapeutic advantage against radioresistant tumours, though it complicates normal tissue sparing and demands refined dosimetry. Treatment planning is now a conversation between physics, biology and computation. Monte Carlo dose calculation engines, 4D imaging that accounts for breathing and motion, and adaptive designs that reshuffle plans as tumours and organs move and shrink. Even as medicine and regulation have advanced, gaps persist. We do not yet possess a fast, universally deployable biodosimeter that reads dose and predicted lifetime risk in an instant for each individual. GH2AX foci and certain gene expression signatures give powerful early signals, but they fade or are confounded by infection, inflammation, and inter-individual variability. Genetic differences in DNA repair capacity, in immune responsiveness, and in propensity for fibrosis mean that two people with the same measured sieverts may have different futures. The lifespan study of atomic bomb survivors remains the gold standard for population risk estimates, but the extrapolation to low doses, to children, to people with comorbidities, and to exposures of mixed quality, internal plus external mixed isotopes, remains an active field where uncertainty refuses to be shaved down to zero. And so science presses forward into frontiers both quietly technical and startling in promise. Single-cell genomics and transcriptomics can, in principle, identify rare cells with catastrophic mutations or unusual responses immediately after exposure. High-throughput qPCR and RNA sequencing panels are being evaluated as rapid biodosimeters that could triage thousands of people within days. Advanced imaging, combining functional PET tracers with anatomical MRI, can visualize inflammation and tissue remodeling before structural damage is plain. Computational models couple Monte Carlo physics with tissue reaction kinetics and machine learning to produce individualized predictive maps, not prophecy, but informed probabilistic landscapes that can guide whether a patient receives aggressive intervention, conservative observation, or a specific mitigator. On the therapeutic side, radioprotectors and radiomitigators are being refined. Amifostine has clinical use as a cytoprotectant in certain chemoradiation contexts. Other small molecules, mitochondria-targeted antioxidants, and agents that modulate DNA repair or inflammatory signaling are in various stages of trial. For internal contamination, decorporation chemistry is better than it was in the mid-20th century, but logistical and biological constraints remain. If policy is the shape of society's answer to these scientific truths, then the modern frame is one of precaution, preparation, and pragmatism. International bodies, ICRP, IAEA, WHO, provide guidance that nations translate into evacuation zones, stockpiles of antidotes, and public messaging. Occupational regulators in many countries have settled on thresholds intended to balance utility and risk. For workers, a higher controlled exposure ceiling, for the public a much lower acceptable yearly limit. Emergency response now anticipates mass triage, combining rapid physical sensors and biological assays to divide people into those needing immediate hospitalization, those who will benefit from outpatient monitoring, and those whose anxieties will need different forms of care. A sober recognition now threads policy debates. Radiation harms are not merely physical, they are social. The panic triggered by poorly explained doses can be more injurious than the exposure itself in terms of health disruption, economic cost and long-term well-being. And yet the most interesting work is intimate and slow. Researchers trying to read the whisper of seconds into the grammar of decades. They ask how a clustered double-strand break turns into a driver mutation that will, 7 or 12 or 30 years later, produce a malignancy. They map the inflammatory milieu left in tissues after cell death that creates a fertile niche for misbehaving clones. They study the role of senescent cells and chronic oxidative stress as intermediaries. They attend to the social determinants that modulate exposure and outcome, nutrition, coexistent disease, access to care, and even prior radiation history. The future will not be a single magic wand that translates an exposure in SV into a headline prognosis. It will be a web of sensors, omics, imaging, algorithms, and crucially social systems that can deliver preventive and therapeutic measures when they will matter most. So in the quiet nights when the mind wanders through the geometry of risk, it is worth remembering the practical moral of measurement. We needed the grey to know how much energy crossed a barrier. We needed the civet to weigh that energy against living tissue and to ask not just how harshly, but how harmfully. We needed microdosimetry to understand that energy delivery at the scale of DNA matters more than bulk averages. We needed history to teach us that instantaneous carnage and slow drips are different beasts that call for different responses, and we needed policy and medicine to translate that knowledge into evacuation maps, into iodide pills handed out at a school gymnasium, into the decision to fractionate a dose across weeks so a child's salivary glands might be spared. The path forward is patient and peculiar. Better real-time biomarkers to triage in the first hours, improved decorporation chemistries, radioprotectors that are safe enough to give broadly predictive models that remember the individual rather than only the population, and a social capacity to act with both speed and restraint when the invisible touches the visible. There will always be humility in the face of what radiation can do, second by second. It rearranges atoms in the space of a breath and sets in motion processes that will declare themselves only in years. History has taught us how to measure that first breath and how to read its consequences. Science and policy continue slowly and carefully to close the gap between a physical event on the scale of femtoseconds and a public health outcome measured across lifetimes. Sleep with that, not with fear, but with the calm knowledge that the ledger exists, that people have learned to count, and that the counting itself has changed the way we build hospitals, write laws, and design machines to aim energy where it heals and away from what we want to save.